So good morning, everybody. Thank you again for this invitation. It's a real pleasure to be here. Thank you, Nagia. We had a wonderful day of consultation yesterday in the school. And thank you to Eduardo Tizzano because I will talk about genetics and this lecture was really important to understand. So I will stick on the time. I have only one disclosure. I am an adult neurologist, but I trained also in early onset myopathy. And I am part of this reference center of neuromuscular disorder, a multidisciplinary center. This is the key how we can diagnose and treat neuromuscular disorder. We are located in Paris region, in this hospital that is in Raymond d'Or, and we are very active in research. We have a big team of researchers where we do skeletal muscle biology, translational medicine toward the neuromuscular disorder. And in our team, we have a tripartite mission. We want to characterize the patient, evaluate them, discover novel disease and mechanism, and then modeling therapeutics. So let's go into this wonderful topic. I did my PhD thesis on congenital myopathy, so I'm very happy. I will do a revision of the general feature of these two entities, congenital myopathy and congenital muscular dystrophy. We will talk about the diagnosis. I will give you some example of novel genes that we need to validate, and then we will go into a therapeutic approach. Not to forget the standard of cares that are very important for our patient. So these entities can present very early in life in a similar ways, but we will see that they are different. And the first difference were noted with histopathology that demonstrated that in myopathies we had a structural change of the muscle fibers, while in dystrophy, the name tells it, you have a dystrophic degeneration of the muscle fiber. The congenital myopathies are rare disorders. They are early onset, but we will see we can have some variation. They are inherited with autosomal recessive, dominant, X-linked or de novo pattern. They usually have normal CK that distinguish from the dystrophy, and they usually do not present central nervous system involvement. They are considered non-progressive, slowly progressive clinical course, but in fact we see that we have a huge phenotypic variability with cases that onset very early antenatally with arthrograde poses, reduced fetal movements, other cases that manifest with motor delays, and then you have a case that can conserve the gait, for example, like this patient, but they have a thin muscle back and weakness. And then you have these other cases that are very mild, but you need to diagnose them. The genetics is complicated because we have now more than 40 genes with wide clinical spectra, histopathologic overlaps, and a complex relationship between each congenital myopathy. The four major groups that have been described through the muscle biopsy analysis are the core myopathies, the Rhodes myopathy, this fuchsinophilus inclusion that you can see here at the Gomori trichome, and then the nuclear internalized that are centrally located in this centronuclear myopathy, and then you have this congenital fiber type disproportion. Thanks to the genetic approach, what we found that now associated with core myopathies, we have 10 different genes. I will only describe the more frequent one. We have Nemaline myopathies associated with 15 genes. We have the centronuclear myopathy, 7 genes. Congenital fiber type disproportion, 11 genes, and some are overlapping. And then we have rare congenital myopathies and myosinopathies. So the prevalence of this disorder is 1.5 for 100,000, and what is you should think of is that there are genes that are recurrent and that are the most frequently encountered in the patient. And for example, this gene, the ryanodine 1, is the most frequent gene of congenital myopathy. Then you have two genes, ACTA1 and nebulin, that are found mainly in rod myopathy. And then you have a dynamin 2 that is associated with centronuclear myopathy. So always think in terms of prevalence when you have your patient, and especially if you don't have the availability of the muscle biopsy in order to classify the patient in different groups. The ryanodine 1 related myopathy, a huge group with different entities that has been described as core myopathies, multi-mini-core, centronuclear myopathy, and they present this alteration inside the muscle fibers that are uneven stained areas with the oxidative staining. Clinically, they can present with a congenital myopathy, but they are allelic of the malignant hyperthermia. So you should advise your patient not to undergo an anesthesia without a previous consultation. And then you have some cases that overlap with the congenital muscular dystrophy with elevated CK level. So this is a huge spectrum of disorder. They are frequent. Think first of this condition when examine a patient with a congenital myopathy. And in our center, we performed a retrospective analysis of muscular, respiratory, myopathologic, and molecular data in 135 adult congenital myopathy patients. And what we found that the rear one, is the last conclusion, was demofrequent gene, and we had patient where the age of onset was considered later than the infancy, the pediatric patient. So you have to think that this condition can express predominantly in adult age. So congenital myopathy in adulthood exists. We need to recognize them. Sometimes they have a gestalt that help you in this recognition. And when you have doubt or the genetic is not so clear, you can do a muscle biopsy. We have some conditions that now are appearing that are very rare but can be part of the congenital myopathy. This is a patient that I saw that presented this tremor. This tremor is not a classic neurologic tremor. It is associated with a mild muscular weakness, and when we registered with the electrophysiopathologist, they told us it's a kind of myogenic tremor. In fact, this condition has been described associated with the gene, the myosin binding protein one gene, with a pattern of inheritance that is autosomal dominant, mild myopathy, and we recently found two sisters from a consanguineous families that presented, sorry for the busy slide, only mild symptoms with hypotonia, with fatigability. We thought for a long time that this patient had a mitochondrial disorder, and in fact in the muscle biopsy there were only some mitochondrial aggregates, and then analyzing them and seeing this tremor, we found that they were mutated in this myosin binding protein gene. So the key message here is also if you have a symptom or a sign that is not classically described in an early onset myopathy, think about it if you have other elements. I don't have to go into the slides, so Eduardo, my homonym, told everything about it, so you know exactly what it is and how we use it in neuromuscular disorder. What is a little bit less, how do you say, rewarding for us, that we have a diagnosis for congenital myopathies in around 40 to 50% of patients. And then you have these views. Eduardo wrote it with the O, I used to use with the U, it's not the problem, it's the same thing. And this is found in the rest of the cases. And sometimes the interpretation is very complicated because you have different gene associated with congenital myopathy. This one, the first one that we will talk about for the therapy, the myopaladin that also we are targeting with the therapy, and the enabling. And so how can you work it and how you choose? Multidisciplinary work and the muscle biopsy. Of course, if you have the possibility to do it, or as our colleagues presented before in the presentation about Lama2-related patient, other system like the skin muscle biopsy. So in some cases we have nice findings that are this candidate gene. One example is what we found in this patient. You don't see the name here, is myosin protein B15, it's written here. This patient had a congenital myopathy with spinal rigidity, core in the muscle biopsy. There was a predominance of type 1 fibers that were also smaller compared to the type 2 fibers, and we did trio exome sequencing in her and the healthy parents, and what we found that there was two variants, one stop variant frame shift here, and another that is a missing localized in the distal part of the gene. So we decided to see if this myosin 15B protein, that is the myosin 15B gene, is a pseudogene, has an expression in the skeletal muscle. We built an antibody that cost us a lot of money, and we did immunohistochemistry and staining with two kinds of antibodies, the C-terminal part of the gene, of the protein, and the N-terminal part. What we found that in the C-terminal part, where the mutation are located, we don't have a similar expression, so possibly there is an alteration going on this myosin 15B in the patient. And then what we found, we found that the Western blot analysis also showed that the protein was reduced, so we thought maybe we are in the good track. And by RT-qPCR analysis, we found also a reduction of some isoform, but not all the isoform of this myosin 15B, which is a little bit misleading. So what we decided to do, we were not convinced. I looked for other patients around the world. Nobody has a patient like this. So in the lab, we set at a point a technique that help to make extraction of the muscle fibers and the nuclei of the muscle fibers, and to sequence them with RNA sequencing. And what we did, we do the analysis on the patient muscle and the control, and we found that the expression profile found in the control in blue and in yellow in the patient was different, because in the patient we didn't find these cells that are the skeletal muscle satellite cell that help regenerating. We confirmed this with immunohistochemistry. Here you have the red dots that are the PAX7 cell. In the patient we did not find, so we can conclude that in this patient we have a reduction of satellite cell that probably is linked to the phenotype, the early onset phenotype of the patient, but we have also an alteration in other genes. What we do next, now we think we have some elements. We cannot validate completely. Why? Because we don't have other patients that present the same phenotype and the same genetic alteration. So we are striving to find other patients and confirm that is the good it. Another example of a candidate gene, this is a patient that I followed from Italy with congenital myopathy. She has some ptosis, she has a prominent scoliosis. You can see here, high arched palate, this lipoma, I don't know, but you have to describe detailed your patient, and she has intellectual disability that do not goes very well with the congenital myopathy. This patient present course in the muscle biopsy that I confirmed by these electron microscopy studies. You can see here the fragmentation of the Z line, and we did the whole body MRI that did not show any pattern similar to other patients, with prominent involvement in all the muscle. Look here, the paravertebral muscle that are completely white, that correspond to the fact that she has a scoliosis. We did exome sequencing. We found two variants in this gene that are present heterozygotally in the parents, and we using these tools of prediction, we found that there's a high probability of a damaging effect of this variant on the protein. That's what to do next. We decided to go back to the muscle biopsy and try to see if this protein was altered. And what we found very interestingly compared to a control, here I only show the patient, that there seems to be an accumulation of the protein in the muscle associated to the area that are the quartz areas. So probably there is a gain effect or gain function of this mutation in this patient. We got more luck than in the present case. Through gene matcher, we found other cases. In total, we have 8 cases associated with the gene, and that was previously described associated with the neuromuscular syndrome, but in this publication, the muscle symptom of the patient, the muscle histology was not characterized. And we are now trying to put ensemble all the clinician and the geneticists to do a publication about it. But to conclude about these 2 examples, so we have the genetics, but we need to functional validate the candidates that we have or the VUS. And how can we do this? Of course, if you have the possibility to have a skeletal muscle, you can do standard histology, and this is already enough. Immunohistochemistry against the protein that are altered, but you can do also more like complex and up-to-date techniques, like, for example, the multi-omics, the single nuclei RNA-seq. But let's go a little bit into therapies that interest us very much. So in congenital myopathy, we have mainly two approaches that have been developed: the gene therapy and the pharmacologic therapy with drug repurposing. There are also robotic approaches, but these are main physical therapist approach that are applicable to other conditions, so I will not go into it. The gene therapy, as you know, is a cutting-edge medical approach. It consists in replacing the gene that is altered in a condition, and the final objective is to restore the normal cellular function and potentially cure the disease. This strategy was applied in a congenital myopathies, the myotubular congenital myopathies, where you have an X-linked recessive mutation in the MTM1 gene that provoke an absence of functional myotubular protein with an altered excitation contraction, and the fact that in the muscle biopsy of the patient you have this centrally located nuclei that correspond to myotubes. What happened? Through a collaboration between the Genethon in France, the group of Anna Bugibello and the Harvard Medical School, they developed preclinical model, mice and dogs, with this alteration, and they treated, okay, with gene therapy that led to the agreement by a pharma, Audentes, to develop a gene therapy for MTM1. This went very well. The dogs were dosed. They did very well because the muscular function was restored. And the first gene therapy clinical trial in humans for X-linked myotubular myopathy started in 2017. We were all very excited, and to be short, what happened that the first data were presented in 2018 with early sign of muscle strength improvement, clinically meaningful improvement in both dose cohort, high dose and low dose. But then there was something that happened. That was the death of 3 children, okay? With signs of liver dysfunction. There was a stop by the FDA, and then other 2 deaths that made that this trial was abandoned. So what happened exactly in this patient? That where we had that both a high dose of the vector, okay, because to transfer a gene you need to do a virus, the adeno-associated virus 9 in this case, but also with the low dose of the vector. In fact, this patient presents a liver pathology that was described previously, but it was outlooked when they went into the clinical trial, because 5 to 10 of patients, they have this peleosis, hepatic peleosis, and what they found, analyzing the data of the clinical trial, that 91 of the participants had an history of hepatobiliary disease. So very important, we have to characterize exactly this patient, because when we inject something that is new, we can have a problem. And AFV-based therapies and hepatotoxicity is now well known, because when we introduce this virus that allow, this vector that allow to deliver the transgene, we have immune responses, inflammation, and damage to liver cell. This can be controlled with immunosuppressant, okay? We have example of successful gene therapies, like for example for SMA, but we need to carefully consider all these kind of possible life-threatening events. So we have now 5 approved AAV gene therapy in Europe and in USA. which interests most us is the Zolgensma for SMA, and now in US there was the approval by the FDA of LFDs for Duchenne muscular dystrophy. Nevertheless, we need to be sure that the immunosuppressive graft can prevent this event, and in the future we will use more efficient IV vector to allow to a dose sparing and giving a smaller dose, avoiding immunoresponse. Where are we now in France with the clinical trial? So Aspiro, that was for the MTM1, is stopped. There was another oligomer antisense therapy for DM2, DNM2 that I didn't detail, that is stopped. We only have these two approaches that are the exoskeleton approaches. What are we doing concerning the therapeutic approach? I have a problem with the progression of the slides. Can you help me to advance? You stop the counter. So what are we doing now? We have a pathology that we described in 2017 that we would like to target with a gene therapy. Okay, it was Caesar. And this pathology is the myopathy congenital myopathy. This is a very rare congenital disorder that is characterized by the presence of these nemaline rods in the muscle biopsy and this CAPS structure. And what we aim to do is to utilize in animal models and in vitro cell models of the derived from the iPS cell of the patient to develop an AFB-based gene strategy. So in the lab we have these mice that present a phenotype that are ablated of the myopaladin. We have this in vitro 3D muscle-derived cell, okay, that we can use and we can inject with the AAV. As myopaladin has a dimension that could enter into this vector AAV, we will construct it and we will try to treat this patient. We are very happy because we got a project financed by the National French Agency for it, and we will go into this approach. Other approaches for congenital myopathy, the pharmacologic therapy with the drug repurposing approach. What is this drug repurposing approach? Before telling you about this, let's talk a little bit about the iPS cell. The iPS cells are the stem cell that we can derive either from a biopsy, either from other tissue of the body, like for example from a blood sample. We can reprogram these cells. Oh, I'm messing everything. We can reprogram this cell and have iPS induced pluripotent cells that resemble embryonic cells that can differentiate in different tissue. In our lab, we have this technology. We can differentiate the cell into the skeletal muscle. And what we do, we got a funding for this MyoPharm project. So we have the patient. We will do iPS reprogrammation. We will phenotype and characterize the cell. I will show you an example. And then we will do screening analysis with the high throughput molecular screening of drugs that have been already approved. So they could be repositioned in order to modify the readouts that we have identified. The congenital myopathy that we are targeting is this disorder. We described it through an international collaboration in 2017. And I follow these patients that now they are adult. And we created from this patient the iPS cell from blood with this particular mutation that you have here. So we have 3 lines of iPS cell. Here you have in the upper panel the control cell. Here you have a patient that has a dominant mutation, the other one the recessive mutation. Both inheritance are associated with the myopathy, the hydropyridine myopathy, which is a protein that they contribute to the relargation of calcium. We characterize these cells, and what we found that we could have cells with a good skeletal muscle differentiation that correspond to the fetal stage of an embryo. So we succeed to reproduce in vitro the skeletal muscle from the patient through reprogramming the iPS cell. And we characterized this. We analyzed, of course, this gene, the HPR, that is localized here. It contributes to the ryanodine that you have seen before, that is also important for calcium handling. And what we found that there is a readout, there is a decrease in the ryanodine, there is decrease in this protein complex, decrease in another protein called Cas-Q1 that could be used for the drug repurposing. We also saw something interesting. Another time, this PAX7 cell, the muscle satellite cell, that seems to be compromised in the patient that you have here compared to the wild type. So we have now readouts. We will go into the pharmacological screening, and maybe next year I will tell you that we have possibly a drug that could be used in this condition and could be translated into a clinical trial. What are the advantages of this kind of approach? We will use only FDA and EMA approved drugs. That means that are potentially safe for the patient. Let's go a little bit rapidly. I have 3 minutes into the congenital muscular dystrophy. You have already seen the difference in terms of muscle biopsy. So this patient present a striking difference. They have a central nervous system involvement. They have increased CKs. Many genes already here, and here just a reminder of the dystrophic pattern with this fibrosis, with this fatty degeneration, and this necrosis that is very prominent. You have two major groups, the CMD with intellectual disabilities, and you can diagnose with a muscle, with a brain MRI associated with a muscle MRI, and then genetic screening, of course. Wolger-Warburg syndrome, muscle, a brain, and this form that was originally described in Japan, the Fukuyamas. And then you have other forms that are more rare, but can overlap genetically to the 3 forms that I example here. And then you have, of course, novel gene. And what you can do if you have a muscle biopsy, you can diagnose a defect in the glycosylation of the alpha-dystroglycan using immunohistochemistry, okay, that orient you, but then you have, of course, to do panel genes or genetics in order to find the hits. And then you have the CMD without intellectual disability. You know everything about the LAMA2 or laminin 211 deficiency that was described brilliantly by our colleague from the Qatari cohort. And then you have other conditions where you can have a secondary deficiency in merosin that you can detect also by immunohistochemistry, that are the dystroglycanopathies that we've seen before. And then you have the lamina C with this drop head. I think that yesterday we have seen one patient with this phenotype. The URIC, we have seen 3 sisters that present a collagenopathy with both hyperlaxity and contracture. And then you have this selenoprotein N that is also classified as a congenital myopathy. It's overlapping with the congenital muscle dystrophy. Where do we stand for the therapeutic approach? I have less to tell. Unfortunately, we are still at an embryonic stage. I hope we are in iPS phase, so we can differentiate. And for pharmacologic therapies, we have some steroid approach in some condition, for example, like laminopathy. And then, of course, the robotic approach. Just 2 examples. In this left minute, there is a promising approach developed in mice that consists to deliver through an AAV mini-agrin in merosin deficient mice. And you can see here that the histology in the mice compared to control is ameliorating a lot with this gene delivery, and also the functional performance of the mice. And the other example is the use of this gapmer antisense oligonucleotide that could suppress the mutant allele in collagenopathies and assure the restoring on the function. And this is particularly interesting because with this approach you can target the 3 genes that are associated with collagenopathies, collagen 6, A1, A2, and 3. So stay tuned because something will arrive soon. Not to forget, multidisciplinary care for congenital myopathy, congenital muscular dystrophy. You have a standard of cares here, nutrition, respiratory care, cardiac workup, orthopedic care, physiotherapy, and thank you very much, I finish in the time. My message are, there are still genetic conditions. So we have, a condition that we need to discover and to describe. And for this, we need a deep clinical phenotype. As Eduardo told, the patient is, all the responses are in the patient. And we need to do functional validation. This is very important, either with muscle, if you have muscle, fibroblast, iPS cell. This is a tools, of course, is expensive, but it's very useful. And then we have a therapeutic approach that are arriving slowly with many drawbacks, but we are getting there, as we did for other disorder, SMA and DMD. And as I told in my last slide, the multidisciplinary care is fundamental to maintain your patient and to be sure that one day they will benefit of the therapies. Thank you very much. This was the wonderful group of yesterday.
We used to know that congenital myopathies are inherited structural defects that characteristic is non-progressive. But sometimes we have patients that showed some improvement with time. Sometimes we have some patients that has later on progressive course. And you actually mention the other form of congenital myopathy. Do you have an explanation for this?
Of the different expression of the same genetic backgrounds.
Why some patient will progress later on?
Yeah, so it's very difficult. We have to think that the skeletal muscle is a very complex system, and we have a differential lesion in different muscles. Some are spared and some are more conserved. There is probably a differential expression of the different isoforms that can explain why some muscles are more affected than others. Then in the congenital myopathies, what you have to think that in some patients with milder form, with the growth, we can compensate, okay? With the muscle gain mass, okay, and can compensate the deficit. That's why some patients ameliorate and can maintain a good level of functionality. But then when they become older, with the aging, unfortunately, they will start to develop a muscle deficit. So I think that the adult onset form are mainly forms that are so mild during the early onset phases that we don't see them, but then with the aging they start developing a muscular weakness. I hope I answered to your question.
Okay. For the stem cell therapy.
For
the stem cell therapy.
Stem cell.
Yeah. How are you going to inject it? Is it systemically, intravenous, or intramuscular?
So these iPS cells differentiated in myotubes serve us only as a model in this drug repurposing analysis to test and to have a model that is not the animal models that sometimes do not respect very much the phenotype, but it's a model exactly derived from the patient, so it is isogenic, okay? It has exactly the same genetic backgrounds of the patient. So it's the perfect model, okay, that respect all the characteristics of the patient. But with this approach, we will not re-inject the iPS cell. This is something that we can do when we genetically modify, we purify it, and then we inject. What we are trying to do is to identify if there are drug compounds approved that can ameliorate the cell and then can be tested in the patient through, for example, other animal models that can be used for the toxicity study. Okay?
Thank you.
The adult onset, excuse me?
Yeah.
اتفضلي يا دكتور. One simple question, the adult onset of congenital myopathy, at what age can you pick up this form of myopathy?
At what age?
At what age you can pick up this form of congenital myopathy, the adult form?
The adult form. So in our study, it was in their 40s. In their 40s that the patient attended to the clinic, and it showed sign of muscular weakness, and so we posed the diagnostic of a congenital myopathy through genetics or muscle biopsy or combined.
One more question.
Yeah, okay. Thank you so much for your wonderful lecture. I have a question. I may have missed it. When you were talking about the induced pluripotent stem cells creation for the DHBR, how did you make the cells succeed to form? Is it through the CRISPR editing or different technology?
So the cell from the patient, okay, that are the peripheral circulating lymphocytes, okay, are then they have already the genetic mutation in the HPR, okay. What we do, we differentiate this, so we put them at embryonic stage, pluripotent stage, and then we differentiate another time in muscle tissue, introducing myogenin, myod that are factors that can induce the muscle differentiation. And what we use as control, this is true, I didn't tell, we will modify the patient from the cell with CRISPR-Cas9 in order to have a wild type isogenic cell that can be used as control. So we will have the muscle undifferentiated and redifferentiated of the patient with the same cell that are devoid of the mutation thanks to the CRISPR-Cas9 system. So we are in the bona fide perfect condition in order to see if there are alterations that we can correct.
So a new idea. Very nice. Thank you.
Thank you so much.